Okay, we've got a few more people in there, so I will begin. Good morning. Oh, I muted myself. We had a few more people join, so I'll start now. Good morning or good afternoon, everyone. Thank you for joining me today for this webinar on non-fungible antibodies. My name is Monique Seymour, and I'm a scientific sales executive at RapidNober. In my role, I work with a variety of scientists to achieve their research goals, either by trying to solve some of the protein and antibody problems they have, or by providing them with tools to further their antibody research. Today, I will be talking about non-fungible antibodies, what they actually are, and their implications as well as applications in our industry. I do apologize in advance if you hear any noise in the background. I live off a busy road, so you may get some zooming cars in the background. So what you're looking at here isn't just a bunch of random pictures put together in a slide. It's actually a piece of artwork created by the visual artist People. It's entitled 5,000 Hours, and it involves 5,000 pictures taken each hour for 5,000 hours, capturing someone's life. It is a digital piece of artwork, and it's also the 6th most expensive piece of artwork by a living author. It was auctioned for 69 million in a matter of minutes. Now, what makes this artwork unique, other than its crazy price tag, is the fact that it's a non-fungible token or an NFT. And the fact that it's an NFT is what really enabled this piece of artwork to hold the monetary value that it currently has. Now, some of you may be going, what is a non-fungible token and how has it enabled this piece of art to be worth 69 million? Well, with the help of blockchain technology, the original version of this piece of artwork has a unique digital stamp that certifies its originality. It's much like having the original Mona Lisa. In today's world, instead of looking at the paint strokes or the actual paint to determine the physical authenticity of the physical painting, you can look at the coding behind the NFT to prove its originality. The concept was first created in 2014 by Anil Dash and Kevin McCoy, who partnered up to draft a code that could be used to certify digital art. It was created as a method to protect digital artwork and provide artists with more control of their work. NFTs are secured by Ethereum blockchain, so no one can modify the record of ownership or copy and paste an NFT into existence. Although the concept didn't take off immediately, today, more and more digital artists are reaping the benefits of NFT technology, whose validation secured demand for digital artists. Now, I'm sure some of you are listening in and are thinking that you've shown up accidentally to the wrong webinar talking about certifying digital art, but don't worry, there is a correlation, I promise. This whole idea had me thinking, if we take a step back and look at the concept, what really stands out? To me, it was the concept of non-fungibility and the use of code as a method of certification for non-fungible items. But what exactly is fungibility? Well, according to Merriam-Webster, it's being something such as money or a commodity of such nature that one part or quantity may not be replaced by another equal part or quantity in paying a debt or settling an amount. Now, obviously, this is a more economic definition, and if we apply it to art and other resources, we could look at diamonds or one-of-a-kind baseball cards or the original Mona Lisa. Nothing can replace any of these things, and they're all very unique in their own way. Fungible items would be something like money, $1 for $1, £1 for £1, or something like oil or sweet corn. And now the moment you've been waiting for, let's take a look at non-fungible tokens and your research. Every time we produce an antibody, do immunizations, do harvesting, all of that is accumulating mutations effects, and different post-translational modifications. Although they may act similarly, the antibody isn't the same, and those differences may not be easy to find unless you actively go after those differences. Much like the Mona Lisa or the piece of art you saw earlier called 5,000 Hours, our antibodies have extremely unique properties that even when they're produced for this purpose of creating more of the same, there are often differences. Most processes look to see if antibodies are acting the same, but they don't actively take steps to determine if there are differences. So here we have a rather artistic image of an antibody. We know it's an antibody because of its overall shape, structure, and its perceived bonds. There is also an added component to this antibody, and it's the colored squares that make it up. If we relate this back to a real-life antibody, we can think of these squares as the sequence. Now, this is also an antibody. Very similar, but with slight differences. We can see that what they're made up of, or the squares, the code, is what makes them different. From far away, we could assume that the potential activity of these antibodies are the same, but upon closer inspection, because we know what the colors of the code are, we know that there could actually be differences in activity. It's the colors and the code that lead us to believe that these antibodies are non-fungible. Now, within the industry, there are ways for us to look at the uniqueness of an antibody in a more comprehensive manner, and that would be the publications that I've listed. Research publications represent an additional source of validation proof for commercially available antibodies. And as such, many scientists rely on publication references to decide on which commercial antibodies to pursue. There are several independent efforts that exist to compile all this information. An example of this is a resource research identifier, also known as an RRID. Now, these numbers are assigned to reagents, antibodies, and publications to help increase transparency. It also allows researchers to keep track of antibodies across academic clubs and industry manufacturers. This is also really useful for when the company or acquisitions there are mergers. The name of the antibody could change, but the RRID remains the same. Although RRIDs do exist, unfortunately, they are not integrated amongst the science community, nor is it used in most databases that are listed here. The antibody registry is the only official database that includes the resource research identifier. Although RRIDs may seem to be an amazing naming convention, a problem with the RRID is that the catalog number continues to migrate as batches change. I'm sure many of you have experienced antibody activity changes when you buy antibodies from different batches or suppliers. So although the RRID is saying that it's the same antibody across different naming conventions, there could still be variations within the RRID. Now, I've mentioned antibodies' uniqueness quite a bit, so I want to actually take the time to go into detail about what makes an antibody unique or non-fungible. We have changes in DNA production, as well as the environment in which the antibody is being created, variations in cell lines, and a wide variety of post-translational modifications. I've separated these into 4 different areas, but that by no means symbolizes that they are exclusive. Many of the comments here are very much interchangeable, and there's most definitely a direct link between protein and DNA modifications and biological and controlled environments. So naturally, when you're dealing with DNA, genetic drift is inevitable. If you have quite a few batches of antibodies in the freezer for a long period of time, chances are there's been genetic drift or even degradation if the antibody often goes in and out of the freezer. Additionally, there can be a wide variety of environmental mutations. It could be caused from travel or interaction with other biological elements. Based on changes from the environment, the energy and natural resting states of proteins are very much dependent on things around them, whether that be a controlled production environment in which temperature, pH, and reducing conditions all contribute interchangeably to the antibody, or variations in biological environments like cell lines. HEC293, E. coli, and yeast all have their own mechanisms for translation, transcription, and ultimately the concentration of the protein that is produced. An antibody produced in HEC293 is going to be different from an antibody produced in E. coli. Regardless of the controls put in place to produce the same antibody, nature does have a mind of its own. Everything I've mentioned also ties very closely to post-translational modifications, as there are so many variables that can cause one post-translational modification. Biophysical interactions that occur in this type of cell line, the physical environment, the production mechanisms, the stability of the DNA, and even the DNA's environment. If we zoom into the antibody, we can see just from this image how many types of post-translational modifications and unique characteristics there are, and this is by no means exhaustive. Deamidation, isomerizations, glycosylation, glycation, this can all happen in many parts of the antibody. Most of these are not even captured or observed by looking at the DNA sequence. All of these small variations ultimately contribute to an antibody's uniqueness. It's only by looking at the end results, the actual protein sequence of the produced antibody, can we actually determine if what we've produced is what we thought we were producing. And this can be achieved through next generation protein sequencing. So further looking at the production of non-fungible antibodies, a study was done to profile IgG1 producing CHO cells to investigate and understand the effects of bioprocessing conditions on productivity and product quality. Shifts in temperature, pH, and dissolved oxygen concentration were found to result in altered productivity as well as product quality, all which contribute to the final biological molecule and ultimately its uniqueness. In one part of the study, differences in deuterium uptake were determined as it correlates with changes of high order protein structure, which could ultimately impact safety, potency, and efficacy of the therapeutic molecule. In the image here, mass spectrometry reveals changes in the amino acid sequence, leading to a possible structural change in 7 peptides of the produced monoclonal antibody. And in this image, we see antibody variants that resulted from variations in temperature, deuterium uptake, and pH, resulting in differences in binding affinity. These antibodies were all supposed to be the same, but changes in environment and production resulted in impacts to the cell line process and changes in post-translational modifications, resulting in shifted amino acid sequence and antibody activity. Now, glycation is a post-translational modification that has very large contributions to the activity and affinity of an antibody. It's been observed in manufacturing processes, and not only does it increase the heterogeneity of antibodies, but it affects product safety as well as efficacy. In the study you see here, non-glycated and glycated fractions were characterized using a variety of biochemical, biophysical, and biological assays. Their goal was to understand the effects of glycation on the structure, function, and ultimately the uniqueness of the antibody. Now, in the image at the bottom of the screen, we can see a correlation of antigen binding with the total glycation measured by a mass spec. Even when the glycation was found on the heavy chain, which is thought to have minimal impact, binding still continues to decrease. So we can easily conclude that characterizing and understanding the post-translational modifications are really very important to truly understand antibody function. Intact, LC-MS, peptide mapping, and next generation protein sequencing are able to quantify glycation levels here. So based on the information we've just gone through and discussing what attributes contribute to an antibody's non-fungibility, the amino acid sequence should really be used as a way to catalog an antibody's uniqueness, from genetic drift of DNA, variations in cell lines and production, post-translational modifications, and finally environmental factors. It's very clear that there is a lot of variety out there, even amongst antibodies that are supposed to be the exact same. The only true way to differentiate between one and the other is to look at the antibody itself, the amino acid sequence, the code. Now, to provide some background on my work with RapidNober in this field, we're a mass spectrometry technology and services company located in Canada, specializing in proteomics and more specifically, next generation protein sequencing, which I will get into a little bit later. We are experts in our field, and we've sequenced over 4,000 monoclonal antibodies to help facilitate characterization, discovery, and patenting. We've worked with hundreds of companies worldwide, and the reason why we're so good at what we do is because we have our own proprietary technology and software, which we continue to improve. And we also have our amazing specialists that are continuously innovating and are hyper-focused on quality. Now, I've talked a lot about the use of code as a way to validate antibodies. And when I speak about code, I'm talking about the amino acid sequence. But I do want to make it clear that I'm by no means saying that DNA sequencing isn't useful or that it shouldn't be used. It's most definitely a great tool, and it should be a starting point for a lot of research, but it's not the only tool. And protein sequencing can be used complementarily as a way to catalog an antibody's uniqueness. If we compare DNA versus the amino acid sequence in terms of the overall accuracy, the amino acid sequence provides a much higher level of confidence. If we take a deep dive looking at the actual code of DNA and amino acids, we can see that one does not directly transfer to the other, even though it's called translation. Despite there being over 20,000 human genes, there is not an equal output of proteins. The 20,000 human genes are translated into millions of proteoforms due to post-translational modifications, alternative splicing, and germline variants, which is why the amino acid sequence detailing the actual protein is so important. Now, I've mentioned next generation protein sequencing a few times, so I wanted to include a brief outline of what it is, the services RapidNober offers, and its application in the industry to help researchers understand and make full use of their antibodies' uniqueness. So first off, we have Repav. This service is based on a technology that captures monoclonal antibodies directly from a subject's blood sample, which gives you direct access to the functional native molecules and the ability to produce recombinant antibodies. Our Remab service provides the full amino acid sequence of a monoclonal antibody. It was our first service offering and what we built off of to create our newer ones like Repav. We have Matchmap, which doesn't use next generation protein sequencing, but it does use associated proprietary algorithms to analyze a protein and determine that its protein sequence matches the expected sequence. Now, with all of these solutions, speed, reliability, and quality are at the forefront of the values that we have when doing this work to ensure that we provide the most accurate sequencing information to enable antibody-centric research. Now, we've worked with a variety of scientists, but specific applications of our service in the context of understanding non-fungible antibodies and requiring the unique sequence have really fallen under these 5 applications, which include discovery and development, IP, regulatory approval, biomanufacturing, and validation and standardization. So first off, we have discovery and development. Our technology has helped enable antibody engineering for humanization. Typically, biopharmaceutical developers would rely on a hybridoma generation or phage display to develop humanized antibodies against antigens of interest. However, homologous monoclonal antibodies originating from other species may already be characterized and commercially available. The sequence of both commercially available or in-house generated anti-animal antibodies can be obtained with 100% accuracy and coverage using Remab. Now, with the knowledge of the primary structure, different versions of the antibody could be generated to suit research need, whether that be to engineer a chimeric version or to tweak areas to make it a better therapeutic candidate. In regards to antibody fragment engineering or alternate variants, such as bispecific antibodies or diabodies, the selection screening process that usually relies on hybridoma phage display can be quite tedious and inefficient because of the massive number of choices for fragment combinations. If next generation protein sequencing is applied here, it could be used to generate a series of proof of concept antibody fragment candidates, which can determine optimal target combinations. Implementing de novo protein sequencing into a CAR-T development workflow allows researchers to easily modify and create many CAR-T models by swapping the antigen recognition domain for variable region sequences found in monoclonal antibodies of interest. This allows for the selection of the best single-chain variable fragment candidates, as well as simple engineering of those regions and detailed optimization of CAR-T models, achieving both enhanced target recognition and reduced non-tumor toxicity. Now, we were the first in the industry to do this using only proteomics. And with our polyclonal sequencing technology, we were actually able to extract monoclonal antibodies from a protein-rich polyclonal mixture for a client that was focused on diagnostic development. Our clients also routinely use our approach in sequencing technology to develop reagents for commercial use, as well as in-house process development. Now, without a secure stream of validated antibodies, developers risk non-specific binding and batch variability, or even a loss of supply, resulting in wasted time, resources, or worse, project abandonment. Before I continue with applications, I'm just going to use the next few slides to break down our own service offerings so that you have a bit more context regarding our applications. So this is our Repav workflow. Now, polyclonal antibodies are really difficult to characterize. Protein sequencing is useful here as you can extract the amino acid sequence from the protein and know whether there are post-translational modifications, which can help with structural characterization. So you're better equipped to deal with polyclonal antibodies. In terms of how we do polyclonal sequencing, our workflow begins with antigen inoculation. So 3 different bleeds are performed and routine purification is used to extract the IgGs. The antibodies are then affinity purified with antigen-specific purification to enrich for the target antibodies. At this point, we use our de novo protein sequencing technology to assess the sample, and it can be used alongside our deep sequencing RNA-seq and NGS data analysis, or we can just go ahead with the proteomics data. So at the end of that, we're able to deliver full-length antigen-specific IgG sequences, which include heavy and light chain pairing. We can also go ahead and perform a comparative ELISA in-house and deliver recombinant target antibodies. We work with clients at different stages of their platforms. So really that means that our clients have the capability to do the immunization or synthesis themselves, in which case we'd be happy to focus on the sequencing side of things as well. So as I mentioned, monoclonal sequencing is often used in discovery and development. So I'll break down how we complete it. When we receive your sample in our lab, the first thing that we do is determine purity. Now you see the small SDS gel there on the left. In a perfect world, when we run it, we would see two bands portraying the heavy and light chains, as the antibody is seen in the middle column there. But often what we see looks like the column to the right, which is the blur that you're looking at, the result of contaminants like BSA antibodies or just noise. Earlier on, that might have been difficult for us to overcome, but after completing over 4,000 sequencing projects, we've been forced to develop technology that allows us to purify these samples so that we can maintain our high quality standards. Now, once we've okayed the purity of the sample, we go ahead and we do multi-enzyme digests. So for context, many people that do next generation sequencing will use between 1 to 3 enzymes here. We actually go ahead and use between 5 to 10. And the reason why we do this is because, well, we've worked with a lot of different antibodies, and we found that our little protease cocktail, as we like to call it, really enables us to have a more holistic view of the protein sequence when it's run through our mass spec. We've also developed unique fragmentation techniques with the mass spec that enable us to better determine between variations of amino acids. So to determine the order of the amino acids, we have proprietary machine learning algorithms, which we like to call novo. We'll do a first draft of the sequence, and our bioinformatics specialists get involved to ensure that all areas have sufficient coverage and meet our quality standards. For context, 20-fold coverage of the sequence is quite standard in our industry, and that's around 95% confidence. We aim for 30-fold coverage of the sequence, which is 99-point-something% confidence. In terms of deliverables, we'll lay out the amino acid sequence for you and highlight the CDR regions as well as any post-translation modifications that we've observed. What you see on the far right is our peptide viewer, where we detail all the peptide coverage and the enzymes that were used. So back to the applications. We've helped clients with their IP applications by providing them with amino acid sequence and aiding in overall characterization. These patents have been useful for a variety of therapeutics, including T-cell targeting antibodies and bispecifics. The amino acid sequence is globally recognized as a protein's code. There are no ifs, ands, or buts, or variations. The amino acid sequence can be legally used to universally protect newly discovered biologics. Generally, the DNA sequence is used for patenting. However, it has been found by a few of our clients that the DNA sequence included in patents did not actually create the antibody of interest. So for regulatory approval, there are two ways in which having the amino acid sequence or verifying amino acid sequence could be helpful. The first is when creating biosimilars. The FDA could require that you match the original biologic. In other words, prove that the biologic is the same unique molecule. Protein sequencing really helps here, as there's some things that intact mass analysis and DNA sequencing can't get. Secondly, our technology is becoming increasingly used to generate anti-drug antibodies, control reagents and assays, testing the clinical efficacy and safety of unique biological drugs. In cases like this, the specific sequence is critical to development of assays, and in many cases, regulatory paperwork may require their exact sequence. With regards to biomanufacturing, Matchmap or Remap can allow scientists to consistently confirm the uniqueness of their antibody throughout the manufacturing process and determine if there are any changes resulting from environmental or biological interactions. So building on the use of Remap and Matchmap for these purposes, a study was done that looked at a variety of different biopharmaceuticals and the maintenance of quality and clinical performance. Cetuximab is a monoclonal antibody which binds and inhibits epidermal growth factor receptor activation. Initial approval was granted in 2004, and commercial manufacturing for the US and Europe occurred in different facilities by different companies. In 2011, It was determined by the FDA that there was 22% higher pharmacokinetic exposure in patients receiving US material. So while a difference in pharmacokinetic exposure doesn't necessarily impact drug safety or efficacy, there is a risk that higher exposure results in clinical differences. This study suggests the use of mass spec to quickly, sensitively, and accurately detect a plethora of protein variants in a single analysis, which could help prevent situations like these. The paper also suggested that mass spec has become an effective tool in manufacturing process development. Next generation protein sequencing can provide further insight for generating product and processes and understanding to inform control strategies and ensure product consistency as well as safety. Now, when we break down antibody validation and standardization, we see specificity and reproducibility. Specificity is generally where the focus is, and reproducibility is typically achieved by repeating steps from a specificity or checking that the sequence remains the same by performing Matchmap. Traditional peptide mapping, liquid chromatography, or the nucleotide sequence. It is worth noting that specificity tests may not address additional chains or progressive changes. It will take many tests repeating specificity to figure out what's going on. If mass spec was used to determine if something was wrong, reproducibility results would be obtained much faster. Now, when validating antibody reproducibility through traditional peptide mapping, you take the sample, complete a digestion, run it through the mass spec, and determine the sequence based on a database. However, you're only looking at the mass, not the sequence, and bias is introduced when looking at mass from other databases. Additionally, because you're looking at other databases, you're also much less likely to catch point mutations. Now, researchers can use Matchmap instead of traditional peptide mapping to have a quick and affordable way of verifying the protein sequence. Given a reference sequence, we basically sequence your protein by performing simple sample digests and running it through the mass spec to very easily determine if the sequence matches the expected or not. There is no bias, and we look at the sequence directly, not just the mass. So to conclude, there are a wide variety of elements that contribute to antibody non-fungibility. And what we've seen is that there is an industry gap in validating antibody originality as well as uniqueness. A solution to this problem would be to use the amino acid sequence with antibody determination and analysis to obtain more confidence in the antibody molecule. Scientists can save a lot of time troubleshooting, developing, and protecting their research. No two antibodies are alike. When you have a good antibody and you're getting good results, you can't replace it. Batch irreproducibility, gene drift, and cell line instability make it very difficult to obtain the exact same molecule. Next generation protein sequencing has helped our clients in all of these situations, and most importantly, in the continuation of their prized antibody. So thank you so much for your time. And I hope I've shown you that antibodies are indeed non-fungible. I'll open the floor to questions.